Hello, my name is Paul Wrezel, and I'm from Regis Technologies in Chicagoland. My presentation today is Selected Case Studies and Impurity Strategies for Drug Substances. This is actually a representation of what I gave at Pittcon 2015. The outline for my talk today will cover impurity thresholds, potentially genotoxic impurities, also known as PGIs, phase appropriateness of specifications, what we see at Regis, as in case studies, and concluding remarks. I'd like to make a couple of comments. First, that the scope of this talk will be drug substances, but many of the principles can be applied to drug products as well. The other comment is that a recurring theme through this will be specifications, which is a very important term because it denotes the limits of acceptance or rejection of a material. So for my first topic, I'm going to cover impurity limits. And the ICH guidelines is a great way to start. They're written for commercial products, but they're often applied to drug substances even earlier in a project as well, such as pre-commercial. Regis is a contract lab, and Regis has a contract lab that supports manufacturing. And we see a lot of customers with many preferences. And some of these customers use the ICH as an inspiration where they'll want their specifications as strict as the commercial guidelines or something less stringent. In either case, these guidelines influence specifications. The guideline known as Q3AR2 has a table in it which covers reporting, ID, and qualification thresholds for maximum daily doses in two different ranges. And obviously, for the higher dosing per day, your thresholds will be more stringent. A number that's often used in conversations is 0.1% limit of impurity for identification, because what happens is that if impurities are greater than this amount, it may be necessary to do extensive work in terms of identification, purification, and qualification in toxicology studies. These impurities typically pertain to the category of impurities known as related substances, which are in turn usually analyzed through chromatographic methods such as HPLC and GC. Sometimes these impurities are now referred to as traditional or non-mutagenic impurities in the context of my next topic, which are the PGIs. This designation is based on structural groups of toxicological concern, or mutagenicity. Mutagenicity will lead to thoughts of whether or not a compound might be carcinogenic as well. There are regulatory guidelines available, which I will present shortly. The class of impurities known as PGIs typically prompts the need for testing with greater sensitivity because of the risks involved. There are softwares available that actually examine structure-activity relationships of compounds when you input structure. And these are great tools for predicting toxicology, especially in terms of potential genotoxicity. One can watch out for big flags like sulfonate esters, anilines, and certain other alert groups. And in any case, determining limits is challenging. Let me show a few tables that will help walk us through this. This first table comes from the FDA guidance, and it shows a relationship between duration of dosing and the amount of impurity that's permitted per day. So going from left to right, the scale goes from short-term dosing to long-term dosing. And I think it makes intuitive sense that as you dose for longer periods of time, the amount of impurity allowed per day would be less and less. This other table is a little busier, but the intention is to just show a few general concepts. It comes from an ICH guideline known as M7, and it categorizes impurities in terms of most of concern, you might say, as in class 1 down to class 5, which is your more or less non-mutagenic or traditional impurity. This might be a simplification, but think of if caffeine was an impurity in your drug substance, it might be like a class 5, whereas something like nitrosoguanidine might be something of a more severe class of lower number. So how you actually define what class is your drug, or excuse me, your impurity in your drug, is a topic for deep discussion, and it's not going to be covered here, but I wanted to make people aware that this information is available. So for my next topic, I would like to talk about phase appropriateness of specifications for impurity levels. So how do you set specifications? And you certainly will use the ICH guidelines as your means for determining specs at commercial stage or even late stage of your project. But organizations that have their act together on impurities and policies will typically have guidelines to cover what is permitted at preclinical and early clinical phases. It's very important that regardless of how you set those specs, you still need to be covered by toxicology qualification data prior to giving any of your drug, and hence impurities in humans. What are possible related substances? Well, there should be an impurity strategy throughout development, and one wants to consider all synthesis, byproduct, and degradation pathways. So what should really be happening in one's project is that all your think tanks, all the talented people in the organization, from chemistry, from regulatory, whoever is knowledgeable about what might be formed during synthesis or degradation, should consider what would your impurity list be. That list could be very large if you start to think of all possibilities. And that's fine, because you could always eliminate things as the project moves forward. What would be a real problem is if you were a deer in the headlights and weren't expecting anything, and then you would be in a surprise mode, and that's never a good thing. Why is that important in turn is that you have knowledge of what might be formed as impurities, so that when impurities actually show up in analyses and there's a need for identification, you have a head start. Impurity specifications tend to be liberal at preclinical and early phases, and they evolve to more stringent by the time you get to commercial phase. I think that's obvious, but it's a point to stress. And of course, the analytical testing rigor relates to that stringency of specifications.